Okay, I think we're probably good to kick off today. So first of all, thank you everyone who has taken the time out of what I'm sure are hectic end of quarter afternoons and mornings to join us today. So this is the second Techspert.io webinar, where we'll be discussing the current challenges we face and can hopefully overcome around the space of cell and gene therapy manufacturing. So I'm joined by a very esteemed contact today, Blair McNeill, who I'll introduce momentarily. But ultimately, what we want to do over the next 45 minutes or so is just understand a little bit about why this space is becoming such a hot topic of discussion, and really what's on the horizon for how we can optimize it, how we can improve, and how we can ultimately scale it with the demands of the industry. So the agenda today is we'll have a brief introduction to the topic and the host. I'll then talk a little bit on ultimately why this is a topic we were so keen to have a conversation about and invite you along to. And there'll also be an opportunity to share some kind of Q&A you might have with Blair here, or any questions you might have around the topic we're discussing. And you can do so, I believe there's a Q&A box probably on the right of your screen. So if you can populate that when you get a chance, I'll keep an eye on that and share questions as they arise. So without further ado, first of all, I have a very brief introduction to myself and Techspert.io. So my name is Graham. I'm the co-founder and managing director here at Techspert. And so we were set up about 6 years ago to try and build a better platform, a better solution to facilitating knowledge exchange globally. And so we started within the healthcare and life sciences industry, really recognizing the importance of accessing high quality, precision matched insights within our industry, especially understanding the rapid evolution of the knowledge landscape within healthcare. I think we'll all agree that the experts in infectious diseases today are probably a little bit different to the ones of kind of 2-3 years ago. And so it's kind of with that goal in mind of accessing better insights, I think puts us in a relatively uniquely and powerful position to identify and now engage with Blair McNeill today. So Blair is head of chemistry, manufacturing controls at Sumitovant Biopharma. And Blair, if I might ask you to give a brief introduction to yourself and your background to the audience.
Sure, thank you, Graham. So my name's Blair McNeill, and I am by formal training, I'm a protein chemist by training and have my PhD in protein chemistry. I've been involved in the process development, clinical and commercial manufacturing of recombinant proteins, monoclonal antibodies for the past 25 or so years. And in the past 4+ years, I've also been involved in the development and clinical manufacturing of gene therapies for our product pipelines. In this activities, I've looked at both the possibility of doing insourcing as well as outsourcing of all our development and manufacturing, our analytics, and helping provide the CMC strategies for our regulatory filings. So hope to bring some of our experiences to the table today, and look forward to the discussion.
Super, thank you, Blair. And I'm sure we'll get into it, but I think the fact that your kind of experience longitudinally spans both the kind of protein and antibody side of manufacturing challenges through to what we now face around cell and gene therapy, I think is probably a fascinating perspective you'll have developed within this space. And so before we dive into the questions, so just to kind of summarize, I guess, as I mentioned, why this is a topic we're interested in speaking on. So kind of for those of you newer to the space, so cell and gene therapy is a combination of modalities which is becoming significantly more common and holds a lot of promise within the biopharmaceutical space today. So for cell therapies, that's about injecting, grafting, administering viable cells into the human body to generate therapeutic effect. And similarly for gene therapy, that's about delivering kind of genetic material for the purpose of genetically modifying a body's own cells, again, for driving a therapeutic effect. Now, as you can imagine, production of such therapeutics is mildly more complex than you might have with a new chemical entity, as we see for small molecule inhibitors, for example, and obviously involves a very complex series of technological processes and regulatory and kind of compliance-based considerations to ensure that the product that comes out the other end of the processing is both safe and effective for administration to patients. And so with these complexities, as you can imagine, the resource investment can be quite significant. And therefore, we come up with some of the challenges we're going to be discussing later on in this session, which I'll run through now. So there's kind of 4 core topics we'd like to visit here. One is recognizing how important and challenging the production of these therapeutics might be. It's about understanding where we see some of those bottlenecks potentially arising, if they haven't already, and what are the kind of innovative approaches that are either currently being explored or might be in the future to try and overcome these challenges. This kind of really comes through to the whole goal here, which is how do we create an efficient and ultimately scalable manufacturing process at the type of demand that we're really going to see within this modality space. For a lot of companies, building internal capabilities is not often the right choice. It's very costly and obviously requires significant time investment as well as cost for the purpose of generating this. So we'll talk a little bit about the considerations a company might make and when they want to engage manufacturing support, and especially when looking with CDMOs, how one might pick those who are best suited for pairing with their therapeutic. And then last but not least, this is an incredibly innovative space, and as you'll see from the challenges, there's a few bottlenecks that, especially as we scale up our demand, that will potentially inhibit our capacity. So we'll talk a little bit about new technologies and new approaches that are potentially going to disrupt this space of cell and gene therapy manufacturing in the future. And then just before I kind of open it up to Blair, I've kind of commented briefly on why this has become such an important topic, but I think gene therapy, for example, isn't necessarily a new modality itself. It was kind of the mid-90s, I think, when it first rose to fame, but then obviously also there were a number of safety issues that led to some challenges of actually scaling this effectively. And I think what we've had to do over the last 20 years is build a more cautious, calculated, and probably ultimately innovative approach to how we manufacture and deliver these therapies to patients at scale. Now, you'll see cell and gene therapy mentioned across the board very regularly, and it's really because what these modalities allow us to do is deliver kind of therapeutics, but treat diseases in ways that traditional modalities, such as monoclonal antibodies or small molecule inhibitors, have not allowed us to access or do. I think this has been especially interesting already within the areas of orphan and rare diseases, where we see previously completely untreatable indications, suddenly there's hope for patients. And with this comes a lot of excitement of the space as a whole. And I think you'll see if you look at the clinical development pipeline right now for either of these modalities, there is a massive uptick in programs being registered, but therefore ultimately in products we hope and expect to come to market, and therefore a manufacturing burden linked to that. So this is why we think today is probably a really pressing and topical time to have this discussion, which obviously very excited to have Blair join us for. So what I'm going to do now is stop sharing my screen, which hopefully worked there, and make myself marginally smaller. But to open things up, Blair, I'd be really interested in hearing your perspectives on how you've seen this field evolve, in particular, what sort of challenges you've seen with the renewed or growing interest within cell and gene therapy manufacturing over the last few years.
Sure. So what we've noticed in the past 2 to 3 years as we've been developing our particular AAV and also lentiviral vectors themselves is combination of materials methods available. We've been co-opting a lot of materials methods that came from laboratory settings and trying to utilize them in a manufacturing setting, which has provided some challenges, both from a quality regulatory standpoint and as well as a scalability, more importantly, standpoint. We've also noticed the advent of assays have really been exploding, both in the details that we can understand, the requirements that we're being asked for, the sensitivity that's needed. These assays are often some of the lagging pieces to a lot of our work as we try to move a product forward through process development and into manufacturing. The other thing, if you kind of view it, kind of step back holistically, is I view the cell and gene therapy kind of where, especially gene therapy, where we were kind of in the mid-90s with monoclonal antibodies is kind of where we are with the production aspects of the viral vectors. Really using what's kind of been out there, and now we're starting to have to think about actually manufacturing, creating bespoke materials methods for gene therapy and can't just rely on what has been used. If we want to take this beyond just kind of what we are today to get to a scalable and more robust process that we'll need for the long-term commercial success of some of these therapies.
Thank you. And so let's say, for example, we zero in on the new assays that need to be developed. Where do you find these are being developed, and what does the process look like for getting these through to kind of industrial production as quick as possible?
A lot of these assays, can you break them down to safety assays, are primarily being developed with CROs, because many of us just outsource those because it's just ultimately a more straightforward way to set it up, run it, than trying to do it in-house or at individual CDMOs. When you get more to the assays that are product specific, they're either being developed in-house if you have laboratories or in partnership with your CDMOs and their analytical development labs. A lot of these are based off of ELISA methods, ddPCR methods, qPCR methods, some of the compendial methods, and HPLC. So these are not necessarily techniques that the CDMOs and yourselves aren't used to running and having operating. Is getting the specificity down for either safety or release of your product to ensure that it is telling you what you need to know and that the regulators will ultimately accept and believe the data from you.
Okay, I think it's interesting you comment on the very important interface and dynamic with regulators through development of such tools, because obviously it's a space where we know there's been a significant amount of regulatory scrutiny, which itself evolves quite rapidly over time. Mm-hmm. So if we go kind of stick with assays for a moment, with respect to kind of assay development, how has one typically engaged with regulators? Is it via the kind of CMOs or CDMOs themselves, or the pharma companies developing assays with kind of guidance by the regulators directly?
The suite of assays we need are kind of somewhat known at this point. There's regulatory documents out there that kind of go through it, and if you're working with CDMOs, the package of assays that are needed are fairly, I'm not going to say routine at this point, but the buckets are known. What each pharma company, biotech company, though, has to do is engage the regulators in like pre-IND meetings where they'd want to put forth the assay package that they're going to use to release their product, as well as the proposed specifications that they would like to see accepted for your phase 1 IND, and get that feedback from the regulators as to what they think. And that was very helpful for us. They brought to attention some newer assays that they want us to run as release assays, like AUC, which before we'd only do for characterization. And then a number of assays that we would have done just for FIO at phase 1, they've actually wanted a set specification, even if it is a wide specification, they want a set specification. And the other one that they really honed in on and wanted to make sure we were addressing was the activity potency assay, and that it followed into the mechanism of action of our drug as best as we could and have that model. They really want to see that the viral vector you make is actually producing an active phenotype at the end of the day. They're not just satisfied with like an ELISA that, yes, is present and not present. Okay. And on viral vector production, so it seems like obviously it's such a core constituent component of how we develop and kind of leverage different properties of effective gene therapies. You mentioned the kind of one of the challenges or areas of significant focus is development of novel viral vectors and production of novel viral vectors. Mm-hmm. So can you comment on kind of some of the key trends or kind of what you see as the opportunities or kind of exciting movements around new viral vector production? Sure. So what we're utilizing now, most people are utilizing triple transient transfection for the initial production of their viral vectors. And there's a number of drawbacks to this. One is it's even when you optimize, it's highly inefficient, and it's not very scalable. Currently, most organizations can only scale to about 200 liters production volume from a bioreactor. And this generally has to do with the instability of the chemical complex of the transfection reagent and the plasmids. There's basically a finite life of that, and it doesn't stay around long enough as a stable complex to go to larger volumes. So in that regard, to be able to scale up, we're actually scaling out and doing multiple 200 liters in parallel to feed into one downstream process. The next thing that to really move that forward into the future, we're going to need to be able to move beyond the transient transfection and into producer cell lines, I believe, where we'll be able to scale up towards that 2,000 liter disposable bioreactor and get more efficiency of scale, economy of scale as we move through. And you take away the inefficiency and the somewhat randomness of transient transfection when we're looking at the production or the expression of the AAVs at this point.
Okay.
And then, go ahead. And then there's just the downstream has its own challenges. In the AAV field right now, there is a affinity resin that a lot of us use in it for the first capture step, which works well. Followed by then we have to figure out how to enrich for full versus empty capsids. And we've kind of a lot of us have fallen into using the monolith type resins. What's interesting has come about in the production of AAVs is a lot of technologies that were developed and thought to be useful in protein therapies like hollow fibers and monolith resins never really gained traction in the recombinant protein monoclonal antibody field, have become very useful in the AAV production field.
When you're talking about the scalability point of, it's not necessarily just about getting a lot more bioreactors, but making them a little bit more efficient. It kind of made me think a little bit of the issue we often have with data centers, where it's trying to get bigger and bigger facilities just to fit more servers in, as opposed to necessarily trying to build something that's more efficient at its core. But on the producer cell lines, which seem like an exciting avenue through which we can achieve more scalable and hopefully through that cost-effective manufacturing improvements. And where would you say this is at? So do we have like set producer lines that are going through regulatory review, or is this something that's still a relatively early stage discussion?
It's still a relatively early stage. There's a couple commercial vendors out there who are putting forth some producers cell line platforms. I think there's going to be some more evolution of those developments, particularly if you want to create your in-house platform versus sourcing one. The biggest part or challenge right now is it takes a lot of time to make these producer cell lines. It can take up to a year or so because you need to have a lot of high throughput screening equipment to be able to look through the various clones, because you're now going through many more clones than you would, say, for a monoclonal antibody producer cell line. So finding basically a cell line that stably expresses your three plasmids at appropriate levels to give you the best viral vector production out of your cells. Obviously, it's a challenge that takes just time, screening, assays, to be able to really find that kind of needle in the haystack, as it were, to really find that cell line that will produce for you at the levels you need and maintain its own stability as you scale up.
Okay, with respect to stability, are there any kind of temperature storage benefits likely to come from producer cell lines versus what we're currently using with transient bioreactors?
No, the stability of the viral vector itself, once it's purified, unless you're incorporating proprietary sequences that lead to a stable production, based on what most people are using for formulation, which is phosphate buffered saline. It's not, minus 70 is kind of the temperature we're freezing at, because it's stable, it gets us there, it works. To have warmer frozen temperatures, or potentially liquid formulation, we really have to invest a lot more time in formulation development, which just hasn't been a priority, because minus 70 works. You take it out, it's a one-time use, you give it, and you're done. So long as your clinical site can handle minus 70, it works today.
Okay, excellent. Just before I go on to the next topic, just a reminder, if anyone has any questions, any areas they'd like us to dive deeper in, please just drop them in the chat and we'll come to them in about 10 minutes or so. I think there's an interesting question as well. So there's probably a lot of earlier stage single asset companies, as well as the larger pharmaceutical companies, who are obviously developing new cell and gene therapy approaches. But as we discussed earlier, may not have the resources to invest in everything being internal with respect to production. So as a result, selection of an external partner to support in this process is not only becoming essential, but also imperative that, I guess, you choose the right partner for the process. So I'd be interested if you could kind of guide us through how you would approach identifying what the correct partner might look like for the purpose of producing your gene therapy, let's say, you're bringing through.
Sure. So the way I approach sourcing an outsourced vendor, obviously first there's just the internet search, that you can find the vendors, many of the vendors who are out there. There's also trade publications that will list them too, a lot of them. Once you contact them and start having that discussion with them about your program, you get an idea if it fits in their facility or not, if it fits in their technical bandwidth or not. Assuming you are comfortable with them and ready to make the next step, it's very important for these programs to go on site and actually talk to the people, talk to the scientists, view the facility, view the laboratories where you're going to be doing this work, and you're going to be spending millions of dollars to do this work. So I went on six or eight different technical business assessments of some of these CDMOs to determine which ones I was most comfortable then going to give an RFP to. So I went from 6 or 8 down to 4, provided RFPs, gave them the opportunity to do the response. And then you do obviously that comparison. And a lot of them, the RFPs, after even you have some of the discussions with them, between all your kind of top vendors, there's not a whole lot of difference in the actual cost structure and the approach. It's fairly similar, and it really comes down to more of your assessment of how you're going to fit in there, what you feel the quality of the system is, both from a regulatory production quality side and just overall general quality. Are you going to get good material out, do you think? Do they have really good technical staff? Or are they just building? Have they been, a lot of them have been smaller viral vector organizations prior to this explosion and were bought by larger CDMO companies. How does that work for you? Has that merger enhanced it or not? That's something you need to evaluate. Does it fit within your timeline? Do they have the analytical capabilities? If they don't have all the analytics in-house, where do they partner or outsource with? One of the other important ones to really work through with the CDMO is the plasmids. Who's going to be responsible for sourcing plasmid? Is it going to be you as a sponsor? You're going to have to go work with another organization, or will they work it for you, or do they have internal capabilities or developing internal capabilities to manufacture the plasmids for their clients themselves. So all these kind of go into that mix of making that decision who you want to work with. We also do some regulatory scouting to see if there were any regulatory incidents, 483s, things of that nature. So those types of things we do also. And then internally too, we have the finance group kind of do a deep dive to make sure that they're a stood up organization and aren't going to disappear on you partway through your production.
Yeah, that'd be a bit of a showstopper, I suspect.
Yeah.
It's interesting, like the importance of the technical case over the commercial case when selecting a partner here, like making sure there is perfect alignment in the capabilities that you need for the specifics, I guess, of your own product. One thing that must be challenging within the space is geographical considerations, because I assume that regulators have a perspective on where you're producing it and what rules you're producing it kind of under. So I'd be interested in how you'd approach a geographical consideration to production.
We're a US-based organization, so we tend to focus for our first clinical trials in the US, and therefore we really look at the FDA and the FDA guidance. However, we are seeing, EU guidance and FDA start to align more. And as we start to develop, we're also looking in an eye of how we're going to move into Europe. So our regulatory strategy is including a lot of that, particularly when you get into the analytics. And that's where we've noticed the main differences is how assays are qualified, the extent of data they want, stability, some of these things. They're a little more regional dependent, and it's really working with the global regulatory groups to understand and get the scientific advice from them, put your plan forward and say, this is what we're going to do. They give you that feedback of they agree or not agree. And as long as you're working a good partnership with them, they frequently will tell you what you really need. This is what we expect. And you follow it, it's great. And if you don't, you're going to have to justify it. And sometimes they agree, and obviously sometimes they don't. But that's been how we've dealt with it from a regulatory. Most of the large CDMOs and the more established CDMOs are used to dealing with multinational regulatory agencies and have set up their systems to be able to handle both FDA, EMEA. Those are the two main ones we're working with these days. So that's how we've mostly been dealing with it. We do know, because we as an organization, we've looked into the Chinese market. We can manufacture viral vector outside of China and import it into China and have we can get through those regulatory hoops. But if you're interested in more the cell therapy piece of it, that actually has to be manufactured within China with a Chinese CDMO. We have run into that in some assets we've looked at. So that makes it a little more challenging.
Okay. And does that barrier, do you find, for example, with China, does that lead to just kind of significant adaptation so it can still be produced and delivered there? Or do you find that people are temporarily excluding the market while they wait for approval in other markets?
Generally, I'm finding, at least ours, is to worry about the Western markets right now, US and Europe, and work through those and understand those. Because once we can understand those, we find that like China and Japan and Australia and some of these others will kind of align themselves and follow suit or take their cues from the FDA and the EU. So it gives us then kind of a better clarity of where we need to go. And then obviously you have to work in the nuances.
Okay, thank you. I have a question from Pascal in the chat, who's interested in your thoughts on how we're currently dealing with or thinking on empty capsids and the issues they present within the space of AAV production.
So our thought is we want to enrich as much as we can for full capsid delivery, because that's ultimately the effective dose you're delivering. It does somewhat depend, we think, on the disease you're going after and how you're administering it. So ours in particular we have a very limited dose that we can deliver, so we need to enrich for as much full capsid as possible to get as much therapeutic dose to the patient as possible. Infusions, you have a little more wiggle because you can put a greater volume into the patient. However, we are finding the FDA is asking us more and more now about empty folds and how are you enriching for those for those folds. And we've been doing it through optimizing the upstream process to get better expression, and then spending quite a bit more time on the second chromatography step in order to enrich for those full capsids. We believe we need the full capsids. The indications we're getting from the regulators on at least our disease state is they want a greater percentage of full capsids over empty capsids. They haven't given us an absolute because they want to see non-clinical information and those types of things, and they'll help guide their judgment on the ultimate human dosage. And there will be a clinical dosing, single ascending dose that we'll have to do to show that the where the effective dose really lies.
Thank you. On that point of kind of evolving regulator considerations, if we were to look forward, let's say 5-10 years or so, are there kind of new asks or new requests or requirements you're expecting to see from regulators with respect to your packages for submission?
We're finding they want more and more safety data, whether it be non-clinical data or analytical data. We're having to do a lot more NGS to do sequence analysis to show that we don't have homologous regions that could lead to a untold event. Again, back to the full versus empty, we're wanting more full capsids there, particularly since right now AAVs are thought to be a single time therapy because of the immunogenic reaction that people have. We're looking into, could we give multiple dose? How could we do it? How far apart would it have to be? What kind of changes to the capsid do we need to make in order to hopefully bring down the immunogenicity, or can we? Those types of things are what we're being asked for. Again, they're asking for more quantitative data, and just more direct data on your characterization of your viral vector. Similar to what you do with an antibody, you have both the biochemical and the biophysical aspects. They're asking for more of the biophysical, some mass spec, AUC, NGS, these types of things, to really understand our final product.
Okay, thank you. Obviously, staying on top of these guidelines is crucial. And I imagine they can have quite a big impact quite early in your development process, when you realize you might have to adapt the vector structure, for example, or the test analytics you're running on it to test purification. How does one effectively stay on top of evolving guidelines?
It's a combination of, there's the official publications, they come out and they're fairly well, if you are staying in tune with the regulatory newsletters that are out there, you kind of know when they're coming out. And obviously it's something you incorporate in. The other, even more effective, is your network of professionals and professional colleagues and friends. Because many of us are using similar methodologies and similar vectors, particularly when you're looking at, as an example, the helper vector. Many of us use the helper vector available from Aldevron. FDA started putting out in meeting requests, in their feedback, concerns about some of the genetic elements within the helper plasmid. We ultimately heard about it through, some consultant friends of ours who said, hey, you may want to consider this in what you're doing. We went through, and this information started coming out, the same language from a number of different colleagues we have at different companies who are using the same helper plasmid, and the FDA was giving us the exact same language. So it was something we knew we had to be aware of, and we went ahead and made changes to our helper plasmid to deal with the concerns that the FDA was having from a safety standpoint, to be able to eliminate that one question. Took us 3-4 months to deal with all that, but it will save us an incredible amount of time, effort, energy, and headache down the road when we actually submit our final package.
There are some very valuable professional contacts, I think, for one to have. Not the product expert, but I think it exemplifies the importance of efficient knowledge exchange for kind of the development and optimization of some of these programs. One question that's also come up in the chat. So we've talked a little bit on how you would identify and when you would outsource production of some of your products. At what point do you think companies should consider making the resource investment for it to be internal? Or what set of capabilities, if you are to have them internal, might one prioritize?
So it's kind of a twofold answer. If you want to do GMP production, you have to want to be able to build that entire infrastructure, and that becomes more of a company decision about the resources they want to put in, because running a GMP facility, you need the quality, you need the supply chain, you need the MS&T, you need the engineering, the maintenance. I mean, you're looking at a pretty hefty investment in infrastructure of capabilities to support the facility, and then maintaining it in a state of compliance all the time. If you're an early stage company, it's not necessarily the best use of money, I would say. The CDMOs, they have that all under control. Go to a good CDMO to do the manufacturing. What you may want to consider bringing in-house earlier, which we are bringing in earlier, is actually the process development piece. Because in any manufacturing process, there's always more and more questions that come up as you start digging into it, getting more data. And you want to be able to answer these questions. Some of them may be small, fairly easy questions. Some of them are more complex questions. You want to be able to answer those fairly expeditiously to incorporate it into your process. It's hard to answer a lot of those questions in a CDMO through their process development. While they have really good scientists, many of them, because they work the way they work through statements of work and very structured experimental plans, they're not real good at flexing when you need to flex because your data takes you in a different direction than you thought it would. And we've started to invest internally in our own process development capabilities to be able to answer a lot of these questions, do these testing we needed to, such as when we went and changed the helper plasmid, and then we also had to make changes to the RepCap plasmid. In order to test those, it was going to take several months at the CDMO and tens of thousands of dollars. We had enough capability already built in-house that we could more quickly test it, do some outsourced testing, and get our answer within about a month. Be able to incorporate that into our process at the CDMO and keep our timelines moving forward. So that's where I would say it's better to make your investment in the kind of development of your platform and technology and understanding than, say, manufacturing itself. And especially if you're a one asset type of organization.
Okay, which I think we are definitely seeing relatively commonly at this point. It seems like it's a bit of a trade-off on yes, there are key advantages in agility if you invest in some of these things internally, but obviously the initial capital expenditure is quite significant, and comes with its own set of risks.
It is, and actually finding people to run it is actually probably one of the bigger challenges these days too. Finding qualified people to run a GMP facility is quite a challenge.
Yeah, that's interesting. We talk about the technological limitations, but there's also a kind of headcount-based limitations within the space experience.
Yep.
So we're coming up towards the end now. So say if anyone has any kind of final questions they want to drop in the chat, please do. Before we get to that, just I know we've spoken about a little throughout this session, but I'm interested in looking at the horizon of cell and gene therapy manufacturing and kind of what technologies, like if you wake up one morning and see something has been proven to kind of, or the regulators accepted, or products got through employing these methodologies into the clinic. So like what new innovations or horizon technologies excite you or have the potential to excite you in the space?
The things that I'm really interested in are the producer cell lines and really being able to bring those to the forefront, particularly for some of our therapies we're looking at that are, while they fall into the rare disease category, there's still a large population of patients that's, it's kind of right on that edge of rare versus not so rare. That really interests me. The other things that really interest me are developing the materials, the methods that are going to be more robust for gene therapy and not just co-opting what we've used in protein therapies. So we do have the affinity resins for AAV. You want to be able to see better methodology for separating fulls versus empties. That's going to be really key, I believe. And then ultimately is some of the formulation development. How do we develop a better formulation for these that may not, so we don't have to necessarily stay at the -70, because that is a challenging supply chain ultimately. AAVs allow, you can get the thawing of the bulk to do the drug product manufacturing. Lenti is much more challenging because it's not amenable to freeze thaw really. So those types of things will make the supply chain easier. Filters are another one. We're using filters based on proteins. We have a lot of loss in those filters due to pore plugging, things of that nature. Can we find better membrane technologies that allow for, that are specific for viral vectors and not, again, not co-opting what's already been out there? Those are the types of things that we're interested kind of from just a practical manufacturing standpoint are kind of the interesting things that I keep my eye on. Okay.
So really interesting kind of probably academic lines of investigation for a lot of these technologies, but if proven effective, could obviously disrupt the space and how we produce these products quite significantly. We're actually just up on time, so we've one question I'm going to try and squeeze in very quickly. If I could ask you to give a kind of 20 to 30 second response, which would just be whether there are any assays or tools to help us predict durability or duration of response of the products we're developing before, I guess, they go in patients.
That's the biggest challenge we're having right now too, is how do we demonstrate durability of ours? I don't have an easy answer for that one. It's a challenge that we're working through right now in our own assay development, and between that and our activity assay are the two that keep us up at night. Okay. Unfortunately.
A problem for everyone. It'd be fascinating to develop, because you can imagine it would quite considerably change the structure of some of the clinical trials we run with these products if we already have a sense on what the kind of durability of response might look like.
Yeah.
So I have just one slide I'm just going to bring up just to close things off in a moment. So first of all, Blair, thank you so much for, sorry, I'm trying to load these slides. One moment. Here we go. Something should pop up. So first of all, Blair, thank you so much for taking the time out of your morning to kind of share your insights and significant expertise within the space. Thank you very much. And also for everyone who's joined today, thank you for taking the time out of your own days to hear Blair talk within these topics. We will send you a recording of this webinar, and we can follow up on any additional questions you might have. But yeah, without further ado, thank you, Blair, for your time.
Thank you very much. And thank you to everyone who joined us today.
All right. I hope you all have a great rest of